This demonstration I call a tale of two acids, and I use this as much to help audiences understand what ocean acidification is not, as I do to help them understand what it is. And I think it's really important to do this, especially when you're speaking to people for whom the concept of ocean acidification is fairly new, because the term acidification, I think, conjures up images in a lot of people's minds of a sizzling, acidic ocean that would be certainly dangerous to swim in and deadly to sea life. And while ocean acidification is certainly a very serious problem, that is not what's occurring. And it's really important for people to have an accurate understanding of what's going on in order to deal with this issue. So this demonstration allows us to compare using a pH indicator dye, which changes color as pH changes. It allows us to compare what happens when you use a very strong acid, in this case lime juice, to acidify the solution versus less acidifying acid, carbon dioxide. And I don't usually say this up front when I'm giving this demonstration. I'm telling you now because you are learning how to conduct this demonstration yourself. But I like to leave a little mystery for my audiences and let them figure out what's going on and predict what they think will happen based on what they already know. It's really useful to use lemons and limes because most people recognize what these are and that they are acidic. Most people have no idea what carbon dioxide does to the pH of water. So that's where the fun lies. Okay, so to get started here, I promised in the, I always promise people that the demos that I teach them can be cheap to perform and can be done almost anywhere. They're very simple, and this one has the added benefit of being non-toxic. Some of you may have seen demonstrations for ocean acidification before where a different pH indicator dye is used than the one I'm going to show you. Frequently, a substance called bromothymol blue is used as a colorimetric indicator, and I don't like to use that one for two reasons. One, it's not that easy to get. You can't go to the grocery store and find bromothymol blue. You usually have to obtain it from a chemistry or biology lab. And two is it's toxic. Actually, there are three reasons, but I'm not going to give away the third reason until the end of this demonstration. So what I use instead is a very simple to prepare solution that I call purple cabbage juice. And I call it that because it's purple, and I make it from the leaves of red cabbage. All you have to do is take a couple of leaves of red cabbage, put them in a saucepan with a cup or two of water, boil it, turn off the heat, and let it steep like a cup of tea. And when it's cooled, just pour the juice off into a container. I sometimes aliquot this out and put those smaller containers in the freezer so I can pull them out whenever I'm doing a demo. And I know it keeps pretty well in the freezer. Not sure how well it keeps in the refrigerator. Anyway, this is our purple cabbage juice. The other things you need for this demonstration are 3 clear glasses, a drinking straw, a pitcher of water, and a source of acetic acid. In this case, today we're using lime. The third thing you need is a helpful volunteer, and it's always fun to ask your audience for volunteers, especially if you're doing this with kids. You get a lot of hands going up in the air. So I call for a volunteer, and they're coming up, and I explain while they're getting themselves in position here that what I have just poured out is regular tap water. You can use seawater for this experiment, but you don't have to. Most tap water is somewhere close to pH 7, which is neutral, and works just fine for this experiment. Now this is my lovely assistant and colleague Nancy, who works at Washington Sea Grant, and she's going to be doing the acidifying for us. So I pour my purple cabbage juice into the water, dilute it in, and I just do that so that I can then distribute it, between the 3 cups fairly evenly. one of these cups of cabbage juice we're not going to touch. That's our control. We need it in order to compare the other samples to. So I'll put the control up here. You can see that it's a lovely purple. Now this, I told the audience the first thing we're going to do is acidify one of these solutions with acetic acid, which is what's in citrus fruit. Lemons and limes work well. I'm going to ask Nancy to squeeze the juice from this lime into the second cup, and then she can give it a stir. And everybody watch carefully. We'll hold this up a little closer to the camera so that you can see. She'll do the stirring up here. So give it a stir, Nancy. Show TV land what's going on. Oh, wow. Wow. Got a really nice color change. And this may actually not come across on video as nicely as it does in real life. When you do this at home, you'll see. So some people have described this color as Barney pink. Remember that stuffed dinosaur? And it really is quite pink. So lemon juice, which is pH 2, quite acidic, causes a dramatic color change. Now people have their expectations. Now you say to them, okay, Nancy, we're going to acidify our third sample with carbon dioxide. I hope you brought yours today.
I hope so.
All right. Everybody has carbon dioxide all the time. It's what you breathe out. It's the waste product of respiration. And I'll have Nancy bend over close to this paper towel because things tend to bubble over. Plus she's wearing really nice white pants. Okay, so while she's bubbling, I usually ask my audience to tell her to stop when they think something has happened. So people are looking for that color change they just saw, and they're not seeing it. And so after a while, because I know Nancy's running out of breath, I say, I think it's time to stop this experiment. We'll see what's going on. Thank you so much, Nancy. And I ask people, what do you see? And it's usually split. People will say, nothing happened, or no, it's lighter, or it's a little more purple. So it's at this point, you want to put it in a row. And again, this is not probably coming across as well on video as it is in real life, but trust me, you will see that the carbon dioxide acidified solution is slightly pinker, headed this way, but nowhere near as pink. It's a little less purple than the original solution. Okay, now you probably think this didn't work, and that's what I always say to my audience. And then I tell them, no, this actually worked perfectly. It shows you exactly what I want you to see about ocean acidification. And that's that when water or seawater is acidified by carbon dioxide, it's not the dramatic 5 order of magnitude, no more, 6 order of magnitude pH change that you might expect based on the term acidification, which sounds sizzling. It's actually a much more subtle change in pH, probably on the order of 0.01, 0.02 pH units. And that's what's going on in our oceans. And even though it's not a big change overall in pH, it is nevertheless a very, very serious problem for marine life. And if you follow up this demonstration with the activity that I call Hold Your Breath, which is about holding your breath, you can prove to your audience members that even a small change in pH, a small decrease in pH in this case, can be very uncomfortable for living organisms, including us. And so I encourage you to watch the next demo and download that protocol as well, so you know how to do that. I think that's all that I want to tell you. If you have any questions about this demonstration, feel free to contact me with the contact information that's included on the protocol, and give me your feedback and your suggestions. Thank you.